Good afternoon, and again, I want to thank Jay and James for the opportunity to speak here and also to visit this beautiful city. I will talk about, I think my work is on engineering side of the cell therapy, and I will use human mesenchymal stem cell to discuss the engineering challenge to translate the cell therapy to clinical applications. So, mesenchymal stem cells are one of the most tested stem cells in cell therapy. And for a couple of reasons. One of them is accessible. And at the beginning also, mainly derived from bone marrow, but very quickly people are able to derive this type of cell from many different tissue types. And you can also expand them pretty well and as a potential to make them as a off-the-shelf cell therapy product. And MSC are excellent source of secretome, including exosome. And these secretome are pro-angiogenic, pro-regenerative, and also in many ways immune modulatory. And so for this reason, MSC has attracted significant potential. At the beginning, people believe MSC has the potential to differentiate, but actually that part of the hypothesis has been proved unlikely. So the cells are currently, the people's interest mainly focus on paracrine effect. That is secreted factors, growth factors, and EVs and exosome that can modulate and help tissue regenerate and grow. One problem is this MSC, a couple of speakers talked about, low engraftment, low survival. So after you transplant for a couple of days, and you will see significant decline in cell number. So how to make MSCs that are functionally equipped at the same time are able to survive in a harsh injury environment and to deliver the growth factors and the secretome in a meaningful fashion? Actually, that's the major challenge. If you look at the clinical study, because MSC has been around for a while, and there are many data to show that a very interesting comparison, that most of the preclinical rodent model, MSC has shown wonderful therapeutic value, and the results are overwhelmingly positive. For smaller clinical trial where MSC are expanded, but are expanded to a limited folds without extensive expansion, MSCs are also, the results are also positive, more positive than negative. For the ones that focusing on producing MSC at an industrial scale and also producing MSC as an off-the-shelf product, which requires expanding the cells to thousands or millions of fold, these results has been overwhelmingly negative. So basically, this shows you that there is a significant problem in producing these cells, and in producing these cells, still maintaining their therapeutic potency. So basically, the one often overlooked property of MSC is one of the early defining feature of MSC is their plastic adherence. But at the same time, this is also the first of major effort in developing the anchorage-dependent cell type for cell therapy. And this means you have to expand these adherent cells millions of fold to design mass production process. And at the same time, MSC has been proven to be highly responsive to environmental cues. So how to maintain their therapeutic significance property in large scale, and also be able to deliver these cells in a meaningful fashion, that's the challenge. And the MSC was originally isolated from bone marrow environment, which is well regulated. We feature very low oxygen tension, extensive ECM proteins. And gradually, after isolating from this environment, they also undergo a significant expansion, oftentimes in a regulated environment, either contacting hard surface or in a on microcarrier or some type of bioreactor in order to achieve the required fold of expansion. How this processing process impact the cell property. That's one of the issues that are impacting the field. And if you look back a couple of years, there were many high-profile studies that demonstrated that MSCs are highly responsive to environmental cues. One of the most well-known is MSC responsive to matrix plasticity. And also geometric cues, nanoscale surface, and mechanical, also have a mechanical memory. This study are wonderful to show that give us deep knowledge on material design and development. At the same time, they also show that this material property highly influence MSC properties. On stiff surfaces, MSC actually undergo pretty rapid change. First is a decline of colony-forming ability, increase in senescence, and a decrease in proliferation potential. And so when we did the initial omics study, among the top GO terms are biological regulation, metabolic process, and also response to stimuli. So in our hands, we wanted to understand, what's the reason that the MSCs are undergoing such rapid change when you grow them in standard culture dish, which is tissue plastics surface. Among the top pathways, the metabolic regulatory pathway, especially glycolysis, pyruvate metabolism, and carbon metabolism, are all upregulated, significantly upregulated in the high passage MSC. So we further analyzed this and then look at, specifically how these pathways are regulated. And if you look at when you pass MSC in the TCP culture plastic surface, the percentage of glycolytic ATP is decreasing. However, the total ATP production from these cells are increasing. So basically, the cells are becoming more active in oxidative phosphorylation. And they also moving from a quiescent state to a more energetic state when you expand them from roughly P5 to P13. And also, at the same time, oxygen OCAR and ECAR are increasing. And interestingly, we also look at mitochondria, and the mitochondrial mass actually increases rapidly when you pass the cells. And at the same time, the mitochondrial potential is declining. And this increase this leads to increased ROS production when the cells are continuously passaged. These metabolic changes leads to a breakdown in cellular homeostasis. So basically, population doubling time increases, so still the cell proliferation rate slows down. At the same time, the cellular senescence also increases, and correspondingly, the stem cell gene expression also decreasing. And also, the cell cycle actually is downregulated. The cells are no longer actively in proliferation state. More interestingly is we also found that there is a basal autophagy also decreasing in high passage MSC. And also mitophagy also significantly decline when the cells are passed to high passages. And at the same time, the genes that are maintaining cellular homeostasis, like a SERT, also decline in high passage. So what this picture tells you is, this data tells you basically is in vitro cellular aging and also reduced staminates. So basically, when you pass the MSCs, and they undergo fairly rapid aging in vitro. If you drive dermal fibroblast through the similar passage, similar expansion history, virtually no change in mitochondrial potential, no change in autophagy, mitophagy, and also senescent properties. So this means this actually MSC, only MSC seems to be going through this decline in cellular homeostasis. So why understanding this? Why understanding the metabolic property of MSC is important? We tested one because MSC are commonly used in immune modulatory therapy. So we tested we tested the response of MSA but after treatment with IFN gamma. Basically, this is immune activation after treating the MSA. And after treat MSA with IFN, you will see that increased glycolytic phenotype. And also, overall total ATP production also increases. So basically, once MSA are immune activated by this cytokine treatment, they enter a more metabolic active state. We further look at whether this metabolic property influence their immune modulatory properties by measuring IDO, which is the main pathway that inhibits T cell proliferation. And we actually found the IDO secretion is depending on the extent of glycolysis. And by titrating using different concentration of glycolysis and the pyruvate, only glycolysis, active glycolysis, sustain IDO secretion in MSC. And this is not only for IDO secretory property mRNA, but at the same time, two other major genes regulating immune response, PGE2 and COX2, are also regulated by MSC metabolism. So when you drive the cells from low passage to high passage, you lost this metabolic plasticity. And this will adversely impact application of these cells in immunotherapy. So the question now is how do we reconfigure the cell metabolism, allow the cell to regain their therapeutic property. Three-dimensional aggregation, which is cell assembly into three-dimensional sphere, has been used in many different scenarios. And one of the early work is the sphere formation use cardiac progenitors to select for the most potent cardiac stem cells. And in MSC world, actually the MSCs aggregate MSA aggregate have also been transplanted in heart surgery, in heart, and has been demonstrated increased engraftment and heart function after injection of this MSA sphere in heart tissue. Higher increased the expression of cardiac markers and also increase the secretome once these cells are transplanted as three-dimensional aggregates. Can you run the So MSC actually can self-assembly on non-adherent surface. And they roughly about overnight, you can see they form spheres, and about 24 hours, they can compact themselves to very well compacted three-dimensional aggregates. So basically, this video shows this process fairly rapidly for overnight. And this has been commonly used in the laboratory. So people use these different laboratory devices to generate these spheres. And one of the, Hung-in job, probably familiar with Hung-in job, centrifuge, or other method, you can make these spheres. But these spheres are not very uniform, or either the throughput is too low. So what we come up with is, can we produce this in a reactor? Can we produce this with well-defined geometry, well-defined morphology, and in a more efficient fashion? There are many different reactors around, and if you look at this process, actually many of these reactors generate environment. You can form one single aggregates, but you cannot form multiple aggregates with controlled size. So in order to really form aggregates, you have to meet two conditions, underflow condition. One is you have to provide enough contact frequency. At the same time, this force cannot be large enough so that you detach the cells from each other. So the process, you basically need to achieve a balance between the aggregation process and also deaggregation, detachment of aggregates. So what we come up with is basically a miniaturized reactor system. So we designed these channels and in different geometric shape. And then we can computational model the level of shear stress. And then by combining this, we can print into a three-dimensional plate and then use this plate to generate aggregates with defined structure. So by modeling, by putting this under wave motion, we can generate aggregates with well-defined structure, with a well-defined three-dimensional structure. This basically shows by adding different cells labeled with different color, you can show that cells are organized into this three-dimensional structure. So the cells derived from aggregates are smaller, they migrate better, and they also have a higher expression of pluripotent genes. And the cells derived from them also has very significantly lower senescence. And also oxygen, but for a long time, the reason that people suspect is because of you create hypoxic zone inside the aggregates, so that's why you have this benefit. But if you measure this, the property of this is actually the aggregate of different sizes all have similar oxygen consumption, and also the oxygen decline is very limited. You do not create a hypoxic zone inside of these three-dimensional aggregates. So what we did is we tried to understand what really regulates this regulation. We again performed the omics analysis, and the metabolic pathway, again, is among the top ones that are being regulated by aggregation. And we used the three populations, bone marrow-derived MSC, adipose-derived MSC, and also dermal fibroblast. And when you do these aggregates, you will see that they basically undergo similar change in biological function. And again, among the top terms that are being regulated is actually EIF2-alpha. This shows that basically what happens during aggregation process is aggregation induce ISR, which basically integrated the stress response. Because of this response, this is a pro-survival and pro-homeostatic response. And this response often triggers two outcomes, or either reestablish homeostasis or induce cell apoptosis. So by aggregation, you trigger this process, and then key pathway regulating this process is phosphorylation of EIF2-alpha. What we found is actually the stem cells has higher upregulation of phosphorylation, whereas a fibroblast response is very limited. So an important outcome of this regulation is reestablishment of the autophagy. So basically what happened in 2D culture is the cells accumulate a lot of dysfunctional organelles, and then they are not able to digest this organelle and gradually become a senescent and aged cells. So by aggregation, actually you can reestablish this ability to basically digest the dysfunctional organelles. So it's time? Yeah, sorry, I do a rapid. So anyway, so that's the aggregate part, and I will just quickly go through this. We don't have a chance to put this aggregates in heart tissue yet, but the model we are working on actually is a stroke model. This basically shows that aggregate-derived cell, once you put it in the mouse brain, the retention time is much longer than the 2D cultured cells. So basically what happened is in vitro, you induce this aging process in MSC, and this decline basically reduces therapeutic potency. Hopefully we can use this 3D aggregation process, rejuvenate the cells, and bring back to their original therapeutic value. Thank you. Yeah.
Thank you very much. Any questions for the following session?
Very valuable data. Thanks very much. But they are, when you isolate them, MSCs are very heterogeneous. At least the mesoblast trials are being done with immunoselected STRO3 expressing cells and with a limited passage to prevent these changes from occurring. Have you tried to look at immunoselected subtypes of these to see whether there are differences between them in these changes you see?
No, we didn't do that analysis. Not the subtypes, yeah.
Okay, thank you very much. In the interest of time, I think we're going to move on to the next speaker. So the next speaker is Tsung Sai.